fragilis. Bacteroides fragilis is a commensal gram-negative, obligate anaerobe that resides in the mammalian lower gut and can profoundly affect the susceptibility of the host to inflammatory diseases. Previous studies have identified B. fragilis as a common opportunistic pathogen in clinical infections and suggested that it may be responsible for a range of diseases involving a permeable intestinal barrier. The scientific name of this bacterium is same of its usual name, Bacteroides fragilis. Characteristics of Bacteroides fragilis: it has a size of 0.5 to 1.5 micrometers to 2.0 to 6.0 micrometers. This is a rod-shaped cells with a rounded ends. This is a gram-negative bacterium, non-motile. This has an outer membrane, a peptidoglycan layer, and a cytoplasmic membrane. This has a capsule and also produces several enzymes. The main byproduct of its anaerobic respiration are acetic acid, isovaleric acid, and succinic acid. This has no pigment or spore formation.
In nutrition, they grow well in minimal defined medium containing glucose, heme, vitamin B12, minerals, bicarbonate, carbon dioxide buffer or NH4CI, and sulfide. This is resistant to penicillin mostly through the production of bilactamase. It metabolizes exopolysaccharides produced by bifidobacteria. Pathogenesis and control. Bacteroides fragilis are prevalent on mucosal surfaces and many produce virulence factors which contribute to pathogenesis. End products of anaerobic metabolism, such as butyrate and succinate, are cytotoxic. Anaerobic or mixed infections result from disruption of mucosal surfaces and the introduction of anaerobes into a normal sterile site. When bacteroides escape the gut, they are responsible for many types of infections and abscesses that can occur all over the body, including the central nervous system, the head, the neck, the chest, the abdomen, the pelvis, the skin, and the soft tissues. Additionally, when they escape the gut, Bacteroides fragilis bacteria can cause dangerous infections. They use human cells as food. Medical or economic importance. Bacteroides species are significant clinical pathogens and are found in most anaerobic infections. Species of the genus Bacteroides have the most antibiotic resistance mechanism and the highest resistance rates of all anaerobic pathogens. It contributes to the maturation of the immune system. In an experimental study, Bacteroides fragilis exerted protective effects of an Clostridium difficile infection mouse model through the modulation of gut microbiota and elevating barrier destruction. They are involved in many important metabolic activities in the human colon, including fermentation of carbohydrates, utilization of nitrogenous substances, and biotransformation of bile acids and other steroids. Most intestinal bacteria are saccharolytic, which means that they obtain carbon and energy by hydrolysis of carbohydrates molecules. Impact into life. Without Bacteroides fragilis, people can have all sorts of health problems. These anaerobes enhance health of the human host by helping catabolize complex molecules such as fucosylated glycans. This helps the body produce immune cells that kill harmful bacteria. They normally help humans in several ways. First, they strengthen the body's defenses by helping the immune system produce enough immune cells to kill harmful invading bacteria. Another important way Bacteroides fragilis Bacteria help humans through their habit of sticking to the gut wall because they take up living space. Bacteroides fragilis bacteria prevent harmful microorganisms from moving in. In addition, Bacteroides fragilis produce substances that help keep the cells of the gut healthy. Human gut cells need these substances to repair and protect themselves.